There we go. I'm starting to see some names coming in. Welcome, welcome. It always takes a few minutes for everyone to get in from the waiting room, so we're just going to hang out. Make sure everything is working for you all. If you have any issues or if you can't hear me or the sound's weird, just throw it in the chat. It looks like we have quite a few people. Beautiful. Okay, I think that's a large majority of people in the waiting room, so we're going to go ahead and get started. Welcome, welcome. So welcome back to those of you who've been here before. I do recognize a few of those names, so welcome back. For those of you who haven't been here for, welcome. We are the Marine Environmental Education Center located on Hollywood Beach, Florida. And unfortunately, right now we are still closed. We're a pretty small facility. We can't socially distance here. So our goal is always to educate whoever wants to learn about a ton of really cool marine science and environmental science topics, give you guys some conservation tools. And since we are closed, we decided to start this really cool webinar series where we reached out to all of our awesome marine science friends who are willing to donate their time and give us a presentation on whatever they are specialized in. So today we are lucky enough to be talking to Briana Coulter. She is currently a Master's of Science candidate at Nova Southeastern University. She came down here after finishing her Bachelor's of Science at Coastal Carolina University in marine science. And right now she is doing a thesis on baleen whales. So she's going to give us a little bit of information about different types of whales, about the baleen whales specifically. And we are going to, oops. Sorry, make sure everyone loaded through. We are going to give you guys a little bit of information about her project specifically. So right now, everyone is muted. Everyone's cameras are off. That is so you can most clearly hear Briana speaking and presenting, and you can see her and her slides. If you do have any questions, comments, concerns, technical issues, make sure you put it into the chat. So I will send a message to the chat so you can all see where it is. You should be able to very easily type it into the chat and we will get to any questions at the end. I will also be here the whole time moderating the chat. So if you have a technical issue or you have something you need answered immediately, I will keep an eye on it. Otherwise, Briana will answer all of the questions at the end. So I think that is about it for my spiel. Whenever you're ready, Briana, feel free to take it away.
Thank you. Hi, everyone. As Taylor said, my name is Briana Coulter, and I am a grad student at Nova Southeastern University, and I am doing my project on baleen whales. So yeah, let's just get right to it. Okay. So whales have been on Earth for over 50 million years, and believe it or not, they actually evolved from land-dwelling mammals. They have a very wide range. They're found in every ocean, and they also have a wide range in size. So talking about whales, so we have the dwarf sperm whale that is 9 feet long and can weigh up to 600 pounds, and that is the smallest whale. And then we can go all the way up to the blue whale, which is 98 feet long and can weigh over 200 tons. And then we have all of the sizes in between. So it's a very wide range. And then if we include dolphins in that mix, we have much smaller porpoises and stuff as well. So there's two main suborders in the order Cetacea, and those include our toothed whales, which is suborder ODONTOCETES or ODONTOCETES, and then also baleen whales, which are in suborder Mycoceti or also called Mysticetes. So looking at ODONTOCETES first, this suborder includes porpoises, dolphins, beaked whales, the beluga, narwhal, and sperm whales. And the main characterization of these whales is the fact that they have teeth. So they feed on larger prey items that are higher up the food chain, including fish, squids, sometimes other marine mammals, sharks, and rays as well. So if we look at the bottom picture down here, this is actually a sperm whale mouth, and the sperm whale is the largest odontocete at about 67 feet long. And if you notice, they actually only have teeth in their bottom jaw. And if you look up at their upper jaw, you can see holes where these teeth go up into because they're several inches long. They're very large teeth. And they actually don't use their teeth to chew like we do. They use their teeth to catch and trap their prey. And they also, so not only do they catch their prey that way, but they also act like antenna when echolocating. So we'll talk about echolocation a little bit later. But just think of the teeth as like a satellite dish on top of your roof collecting the signal. So it collects their echolocation signal and then it receives it and then it transfers it through their lower jaw to their melon, which is very prominent in the beluga. So their melon is a whipped fat that is used for echolocation. It's really good at receiving sound. And if you look at the beluga whale, this round head up here, that's where the melon is located. And the ODONTOCETES are usually smaller than baleen whales. Obviously, the sperm whale is quite large, but typically speaking, they're usually smaller. So now going on to baleen whales. Baleen whales, they're my favorite. So they are filter feeders, and that means that they don't have teeth. So they are adapted to have what's called baleen plates. And you can see it really well in the picture here on the right. And it basically just looks like hair is in their mouth. And that's a pretty good guess because the baleen plates are made up of keratin. And keratin is the same type of structure that our hair is made of, our nails, also fur. So the baleen plates, there's hundreds of baleen plates in their mouth and they are overlapping each other and they grow continuously from their upper jaw. So they don't have ones on their upper and lower jaw like most whales have, like ODONTOCETES have with their teeth, but they are continuously growing just like our hair and our nails are continuously growing. And that's because the bottom is very bristly and it breaks down and it breaks off. So it has to be continuously growing and replacing what's being damaged at the end. They use their baleen plates to filter, sift, and trap much smaller prey than fish or squids. So they are trapping phytoplankton, so the primary producers in your ocean ecosystem, zooplankton, krill, and then small fishes. So most baleen whales have very extensive migratory patterns and they travel long distance to their breeding and foraging grounds. So a good example of this and a well-known track is for humpback whales. So humpback whales, they will spend their winters down in the Caribbean on the east coast of the United States or off the coast of Hawaii or in Mexico on the west coast. And so they spend their winters down there where it's nice and warm. And then whenever it starts to heat up, whenever we get into summer and at the higher elevation, so in Alaska or the New England states, it's a very productive ecosystem, meaning that there's a lot of food, there's a lot of prey for them to eat. So basically, they will start to swim up to the buffet up at the higher latitudes, and they will feed all summer long and they'll get nice and full and happy. And then whenever it starts to cool off and the ecosystem's not as productive anymore, they will swim back down to the warmer waters. So baleen whales, they know what they're doing. They prioritize their temperature. And so due to the wide ranges that they swim, they're in all of the oceans, it's very hard, and they're very large, right? So it's very hard to track them, to study them. But scientists and researchers are getting very creative on ways to study their habits using tags, and then also using their baleen even, which we'll talk about a little bit later. So why filter feed? Why have the adaptation to have baleen plates instead of teeth, right? So like I said, they eat much smaller prey. So phytoplankton, zooplankton, they are on the scale of millimeters to centimeters long. So they're very tiny. And if they had teeth and they went to chomp down on something 2 centimeters long, it would just swim away, right? So they have adapted to have these baleen plates acting like a filter to trap all of the very small prey that they are consuming. And this also allows them to squirt out water so that they're not ingesting a bunch of seawater. So what they will do, there's a couple different modes that we'll talk about, but generally speaking, what they will do is they'll open their mouth wherever the prey is, and they will let the prey and the water come into their mouth. They will then close their mouth. Whenever they do that, they will push their tongue up to the roof of their mouth, and this will push all of the water out through these baleen plates. So you can see how they're stacked here. So all of the water will come out through those plates, but inside it's very rough. So all of the bristles are very rough and spread around. So this traps all of that really small prey. And they'll push all of the water out and all of the prey will be stuck inside. And then they actually use their tongue as a scoop, and then they'll scoop the inside of their mouth, collecting all of that prey that got trapped in there on their baleen plates. So now we'll talk about the different feeding styles. So there's 3 main feeding styles for filter feeders for baleen whales. So first, we're going to talk about the skim filter feeders. So this is an image of a North Atlantic right whale. And you can see that the whale's pretty round, right? It's a pretty round, large whale. And skim feeders, they actually feed horizontally in the water column. So what they will do is they will open their mouth as wide as they can, and they will swim straight through a dense aggregation or an area in the water that has a lot of prey. So a lot of phytoplankton, a lot of zooplankton. So that's the very, very tiny prey. They'll swim right through that, and then they'll close their mouth and do all of the things with their tongue and trap that prey. So because they are feeding on very small prey, they also have the finest baleen fiber. So think of a baleen fiber as one strand of hair. So they have the skinniest strand of hair out of all of them, and that's because they're eating the smallest prey. So then we'll move on to the gulp and lunge feeders, and you can already tell, so this is an image of a blue whale. You can already tell that it's much more streamlined, much skinnier body shape compared to the North Atlantic right whale. And that's because the gulp and lunge feeders are more agile in their feeding techniques. So they actually feed vertically. They will feed horizontally sometimes, but mostly they'll feed vertically in the water column, meaning that they will track smaller fish. So they're catching prey on the scale of inches. They also do centimeters like zooplankton, but it's bigger prey. And so therefore they have slightly larger fibers. So they will find a dense aggregation of fish or zooplankton, and they will jump or lunge into that, opening their mouth and then closing it. And a good example of this is humpback whales. So if you've ever seen the videos of a group of humpback whales coming up out of the water, catching a school of prey, that is an example of your gulp and lunge feeding style. And then lastly, we have the benthic feeders or the suction feeders. And there's actually only one whale that does this, and that is the gray whale, which is pictured here. And the gray whales are unique in that they will swim down to the bottom. So benthic means that they are bottom feeders, and they prey on tube worms, amphipods, which are small, like 2 centimeter long crustaceans down in the sediment. So they have the widest fiber, so it's very coarse, and they actually have a favorite side. So it's like being right-handed or left-handed. They will swim down to the bottom, they'll tilt to one side, they open their mouth, and they will actually depress their tongue, or they'll push their tongue down into the bottom of their mouth, and that creates a suction, and that will suction up the sediment and the prey up into their mouth. And once they have all of that in there, they will close their mouth. And because they have the coarser baleen, they can actually push out not only water, but sediment, because they don't want a lot of sediment in their stomachs. So they'll push out the water and sediment, trapping their benthic prey, and then they will scoop that out with their tongue as well. So those are the 3 main feeding styles of our filter feeders. So looking at mammals in the water. So there are a lot of challenges to being a marine mammal in the ocean that they face. And those challenges include movement, heat, oxygen limitation, and sound. So looking at body structure. So the streamlined body and other adaptations assist whales with cruising the ocean with minimal effort. So the fusiform body shape is the ideal body shape, and that's because it reduces drag. So whenever you're swimming in the ocean, you want to reduce your drag. So then that way it's easier, you're using less energy, and that way you can put energy to other things like reproduction. So if we look at this image down here, it's a very good image showing this fusiform body shape. So you have a very narrow nose, a very narrow front of the animal, and then it will expand and get wider in the middle, and then it'll narrow back out at the end. So you can see we have a very narrow front, it gets wide right here through the middle, and then we taper off and gets narrow at the end. And this just creates a body form that the water just kind of flows off of their body instead of pushing against them. And then they also have smaller limbs to reduce drag. And one way, a good example to use is whenever you are swimming, whenever you dive, you want your hands to be together and you want to be this long streamlined body so that you can go farther in the water. Or if you went into the water and you had all your arms and legs out, you wouldn't go as far, right? So they want smaller limbs as well. So if you look at the dorsal fin up here, you can tell it's much smaller than like a shark dorsal fin, and that is just to help reduce drag. And then the bigger you are, the smaller the volume to surface area ratio you have. And this also just makes them more energy efficient. So they're using less energy to swim. And it also helps with pressure. So pressure increases greatly as you increase in ocean depth. And sperm whales, they're the deep divers, right? So they go all the way down and they hunt squid and other prey items. So this also helps with that as well. So next we're going to talk about heat. So water conducts heat 24 times faster than air, which means that it takes a lot of energy for whales to stay warm. They are mammals, which means they produce their own heat. They're warm-blooded or endothermic. So they have a couple of strategies for staying warm. The first one is blubber, which I'm sure we all know about, right? And blubber is basic, it's not just fat. So there's a lot of connective tissue in blubber, which makes it more insulating, and it gives it other properties so they can store energy in their blubber. So it's not just fat, it's a little bit more than that. And so your skim feeders, they have the rounder body, right? So they're gonna have more blubber. They also are found in colder climates than your whales that are more streamlined, and they don't need as much blubber if they're not in cold waters all the time. So they also have this mechanism called a countercurrent heat exchange, which is demonstrated in this picture here. So warm blood in their arteries, or from their arteries, from their core, goes out to their extremities. So their flippers, their tails, whenever it goes out to their extremities, because the surface area to volume ratio is greater, so they lose more heat from their extremities. It's really easy for them to lose heat. So the blood that's in their extremities gets cold. And then whenever that cold blood coming from the veins, it is actually right in line, it's right beside the warm blood from the arteries. And what happens is the heat transfer between the warm blood from the arteries occurs to the warm from the cold blood in the veins. And this makes it so that the cold blood from the extremities is not as cold. So the blood that's coming from their flippers to their core is not as cold. So it doesn't bring down their core body temperature. So it helps them maintain a constant body temperature. And then they also have the mechanism called vasoconstriction. And vasoconstriction is the ability for them to reduce blood flow to their extremities. So if there's less blood going to the extremities, there's less heat that can be lost that way. So they utilize this for heat, but they also utilize this whenever diving to make so that they can have longer dives. So now we'll move on to breathing. So I don't know about you, but I can only hold my breath for about 30 seconds, and they definitely take the cake. They can hold their breath much longer. So first off, they do have a large lung capacity, so they have much bigger lungs than we do, but they're also just more efficient at their oxygen transfer than we are. So what their lungs can absorb 80 to 90% of the oxygen. They transfer that much in their lung capacity, where we humans only do about 15 to 20%. So they're more efficient and they can transfer their full lung capacity in a matter of seconds, right? Whenever they come up to breathe, it happens in a matter of seconds. Whenever they're diving, they can also reduce their heart rate to use less oxygen and also the vasoconstriction, so they can maximize how long they're down there, since a lot of them dive for their food. And then their nostril position is up on the top of their head instead of front and center like us. And this helps for them to minimize their drag whenever they're coming up to the surface, and it just makes that whole process a lot more efficient. So one interesting fun fact is that the nostrils of whales, whenever they are relaxed, they're closed. So that's different than us, right? Our nostrils are always open. But whenever their muscles are relaxed, that means that their nostrils are closed, and they actually have to flex the muscles to open up their blowhole whenever they come to the surface to breathe. And then lastly, so to communication, we're coming back to echolocation. So sound travels 4 times faster in water than air, and the ocean is very big, right? It all kind of looks the same underwater. So it's easy for them to get lost. Which is why they have modified how they use sound to not only communicate with others, like with other individuals and their families, but also to track and hunt food. So toothed whales, or the odontocetes, they use echolocation to locate their prey. They will produce it's kind of like sonar. So they'll produce the wavelengths and they will send that out. It will hit on their prey. So let's say it's a fish, and it'll bounce back to them. And remember, the sound will come in the antennae of their teeth, and then it'll flow through their lower jaw up to the melon. And then that way they can perceive how far away it is, what's the size of it. They can even determine how many are there. So it's a very detailed image and idea of what they get. And whenever they're further away, they'll send less echolocation signals to determine a general idea, and then the closer they get, they increase in frequency, so they can have more detail to help them capture their prey. So this is primarily used by odontocetes, not seen in mysticetes. Mysticetes, though, they still do communicate. They produce sound to communicate. The best known one are humpback whales. They have very long and intricate songs. They can last, I think it's like up to 15 to 20 minutes long of continuous singing. And they can even identify individuals because their songs are unique, which is really cool. The baleen whales, they typically use lower frequencies, where the odontocetes and echolocation has higher frequency sounds. So let's talk about the threats that face whales. So commercial whaling was banned in 1986, but it has and still is having a drastic impact on whales today. So majority of whale populations were impacted by like 50%, even greater, before the ban, and then it still is occurring. So commercial whaling is still occurring in the oceans today. Also climate change. So climate change is impacting our ocean temperatures, which is impacting primary productivity. And we're seeing, since these whales feed along the bottom, the base of the food chain, on phytoplankton, on zooplankton, because their food is being impacted, we're seeing an impact in the whales as well. Also pollution and environmental pollutants, which I'm going to talk about a little later for my project. And then also noise pollution. So because sound travels so far, and because they rely on sound so much, the oceans are a very noisy place with all of the commercial shipping that happens, sonar, and all of the things that happen in the ocean sound-wise. So this can confuse whales, and it can impact their ability to hear and communicate. Also entanglement in fishing gear. So this is obviously a problem. If they are entangled, they can't come up to the surface and breathe. And then they unfortunately drown. So, and then also boat strikes. So I know there has been efforts for boat strikes because the boating industry harbors also often correlate with common feeding grounds for these whales. So I know there have been efforts about changing boating like pathways so that they're not in high use areas of whales. So there are efforts to try and minimize that. So now I'm going to talk about my research and what I'm doing here at Nova Southeastern University. So my main research question is, can we quantify organic contaminant concentrations from baleen plates? So now I'm talking about the environmental pollutants that are in the water. So what are organic contaminants? Organic contaminants are hazardous chemical compounds that are resistant to environmental biodegradation. So that just means that it's hard for normal environmental processes to break them down, which means they stay in the environment and in the ocean for a long time after they've been used. And then organic, what makes them organic is just carbon and hydrogen bonds that are present in the chemical molecule. So there are a wide range of contaminants, but what the contaminants that I'm primarily looking at and focusing on for my project are polychlorinated biphenyls, or PCBs. There are 209 isomers, and an isomer just means a variation. So there are 209 different types of PCBs. So PCBs are flame retardant chemicals. So they have a wide range of use. They are in paints, they were used in wiring and like plastic coverings. So, but they have a very wide range of use, and they were used primarily from the 1930s until they were banned in the 1970s. Then next, I'm looking at dichlorodiphenyl trichloroethane. That's a long name. So those are DDTs, and DDTs were used as a pesticide until being banned in the 1970s as well. Some countries still reserve the right to use DDT today for malaria control. There are 6 different isomers, so 6 different variations of DDT. DDT is what is primarily used as a pesticide, and then once it gets into the environment, it breaks down and transforms into DDD and DDE. And then lastly, I'm going to be looking at hexachlorocyclohexanes, or HCHs, and those are also a pesticide or an insecticide. And there are 8 different variations of that as well. And it was also primarily used from the 1940s to the 1970s. So contaminants in the marine environment. So the organic contaminants can also be called persistent organic pollutants, or POPs. And most of the use is on land, right? So in our farming. So how does it end up in the ocean? It enters the ocean through air-water gas exchange. So through emissions of these contaminants, they can actually dissolve or enter into the ocean just from them being in the air and coming down into the water. Also through runoff, so in agricultural farming and everything, whenever it rains and there's pesticides being used on those crops, whenever it rains, the rains will pool the organic pollutants into creeks, into rivers, and all of those rivers will most likely enter into the ocean. And then also dumping. So some plants have just dumped their remaining or leftover pollutants into the ocean as well. So those are the 3 main forms of how they enter into the marine environment. So why are contaminants an issue? So contaminants, high concentrations of contaminants can have negative impacts on health of not only animals, but also humans as well. They are lipophilic, which means that they are fat loving. So they will attach to lipid or fat in an organism, and then it will stay there. So let's just say it starts in phytoplankton. So the phytoplankton absorb the contaminant or the PCBs, and that is stored in their fat. So then the zooplankton, they eat that phytoplankton, and they are now not only getting the nutrients from the phytoplankton, but they're also getting the contaminants from the phytoplankton. So they have to eat a lot of phytoplankton. So they are consuming all of the contaminants and storing them in the zooplankton. So then the fish will eat the zooplankton. So now they are consuming all of the contaminants that the zooplankton have. And then this just has a magnifying effect all the way to the top of the food chain. So large mammals typically have the highest concentrations because they are eating at the top of the food chain. So there's all the time throughout the food chain for organic contaminants to accumulate in their bodies. Because they are resistant to degradation, that's why they stay in the environment. So they stay in the environment for phytoplankton to continue consuming them even years after it's been used or years after it's been exposed. And it's hard for the environment to break them down, which means they stay there for a long time. So contaminants specifically in baleen whales. So because baleen whales feed along the base of the food chain, they feed on phytoplankton and zooplankton, they are used as an indicator species on the overall health of an ecosystem. So if we know what type of concentrations are in baleen whales, then we know what type of concentrations are at the base of the food chain for all species to be consuming. There's typically lower concentrations in mysticetes than odontocetes because mysticetes are feeding at the base of the food chain, and odontocetes they're feeding on fish, mammals, so they're feeding higher up on the food chain. So there's more time for the contaminants to accumulate. And then, so why is this important? Baleen whales are hunted by Inuit communities, and they rely on whales for subsistence throughout the year. And it's important to know what type of concentrations are in baleen whales so that we know what type of concentrations people are consuming for their health. So why baleen? There has been previous studies done on organic contaminants in all kinds of animals, but especially in baleen whales. Previous research has included blubber, mussel, and liver. So blubber is a great substance to use because it is lipophilic. So it loves the fat, and so they are predominantly stored in blubber. So it gives you a good representation of what type of contaminants are in the animal. Also with mussel and liver, but the one drawback with using these organs is because it only gives you a snapshot of the contaminants that are in the animal at the time that you sample. So we can get plugs of the blubber or the mussel, so you don't have to injure the animal to get the data, but you have to have a multi-year study to see any type of time scale. So how are these concentrations changing over time? Where baleen is continuously growing. So because baleen is continuously growing, we can get a timeline or a time scale of what the concentrations are. So whenever the baleen grows, it traps what's happening in that animal at the time that it grows. And then we can use other data like stable isotope data to determine where the whale is at, where they're located, and at what time they were there. So then that way we can have an idea of what the organic concentrations are in this time and in this location. And we can compare that over years, especially with the skim feeders. They have the longest baleen, and so therefore they have the longest time scale. So we can get up to 20 years of information from one baleen plate. And then this will also give us the ability to calculate accumulation rates, since we have a known time. And if we can quantify concentrations, we can calculate how fast they are accumulating these organic contaminants. So we are going to be using bowhead whale baleen plates. So bowhead whales, they are skim feeders. So we're going to have a very long time scale. We're going to have 20 years of data. And bowhead whales, they are up in the Arctic Ocean. So they follow the ice pack. Whenever they migrate, they follow the ice pack. So in the winter, they are down in the Bering Sea, and then they will follow the ice as it melts all the way up to the Beaufort Sea in the summer. And then they will spend their fall areas. The stock that we're looking at, they'll spend their fall feeding area right here between the Chukchi and Beaufort Sea. So we're looking at the only stock or population of bowhead whales that is located in US waters, and they are also a stock that's considered endangered. And then here's an image of a bowhead whale as well. So nobody, to our knowledge, nobody has done this before. So nobody has tried to get organic contaminants from baleen plates. So this is a very methodology heavy study. So we're having to try a couple of different things to figure out what method is going to work the most effectively on extracting the organic contaminants from the baleen. So the baleen is going to be shipped to us from Alaska, already powdered and ready to go. So they powder it using a drill. And once we get that, we're going to weigh it. And then we have two different methods. So the first one over here is a digestion. So this instrument is a mod block. And we will put our baleen with nitric acid and hydrogen peroxide in the mod block for 2 hours at 60 degrees Celsius to try and extract the contaminants from that powdered baleen. And then we are also trying an incubation. So this is an incubator over here, and this is the exact one that we have in the lab. And we will put the baleen in there overnight at 40 degrees Celsius with hexane and dichloromethane. So we're testing these two extraction methods. So once we digest our baleen, we will do a liquid-liquid extraction, and this will be the same and consistent for both the digested samples and the incubated samples. So a liquid-liquid extraction, we are essentially going to take our sample with the digested baleen, and then we're going to add our solvent. Our solvent is dichloromethane and hexane, and then we put it into a separatory funnel, which looks just like this. This is the setup that we do, and then we shake it so that we are extracting all of the contaminants from our digested sample. And then we'll get two nice layers that look just like this. And then all of our organic contaminants, all of our POPs are going to be located in this top layer. So we keep the bottom layer just in case, but we are primarily looking at the top layer. Next, after we do a liquid-liquid extraction, we do that twice, and then we will do a solid phase extraction. So this is just to make sure, because we're doing two separate extractions, just to make sure that we are getting the most contaminants possible and we're getting a very clean sample. So this is what our solid phase extraction cartridges look like. And there's just acidified silica in there, which just makes sure everything in the sample is very clean. And then next, we will dilute our samples so that we're super concentrating. Since we are expecting to get low values, we will dilute our samples so that we super concentrate it and we can get all of the contaminants as possible. And then we will load it into a gas chromatography mass spectrometer, or a GCMS. So a GCMS is pictured right here. So this right-hand side, this is the GC, this is the gas chromatography side. And then over here, this is the MS, this is the mass spectrometer. And then we have a column, which looks exactly like this. So this column is essentially very thin glass. And to put into reference, this column is 60 meters long. So it's very thin and it's wrapped around the coil pretty tightly. And we load that into the GC and we will load our sample. Our sample will be put into the column, and we have varying temperature settings that we do. And so we'll hold a temperature for a certain amount of time, or we'll ramp it up really fast. And then this is so that we can get all of our contaminants to come out at an individual unique time. So we want them to come out at a unique time. We call that a retention time, because we need to be able to identify what type of concentration, what contaminant is coming out. If they all come out at the same time, we're not going to know which one's which. So they go through the GC and they exit the column at individual times, and then the mass spectrometer is what's going to read. So it is what identifies what contaminant is coming out at that time, at the retention time. So we do have some results. We don't have any concentration values yet, but we have figured out the GCMS method. So this took us about 2 months to figure out what temperature settings we needed to get all of our contaminants to come out at separate times. So we are looking at 21 different contaminants, and they are all labeled here. So our HCHs, we have alpha, beta, and gamma. Those are the different isomers. They all come out first. And then we have our DDTs right here in the middle. And then we have a wide range of PCBs. So we have some lighter PCBs, 28 and 52. And then we have PCB 209 over here, which is our heaviest one. So all of these peaks, these are all contaminants. These are our standards. So now that we know what times they come out for any of our samples that we run, if we also get a peak in those samples at these same times, we will know that this is PCB 52. So in summary, why should we care? Why do we invest so much time into baleen whale research, into whale research in general, and why are we trying to get so creative on learning more about these animals? And that's because whales are ecosystem engineers. They play a critical role in the overall health of their ecosystem. So it's actually called the whale pump. And basically what it means is that baleen whales, they feed on phytoplankton. Those phytoplankton need nutrients. They need nitrogen. They need all kinds of phosphorus to be able to grow and produce. And the waste or the poop that whales produce in these ecosystems, they have all of those nutrients in there. So those nutrients fuel the phytoplankton. They fuel the base of the food chain to make it sustainable throughout the whole summer. And then phytoplankton not only is the base of the food chain and supports all of the trophic dynamics and animals that live there, but they also release oxygen as a byproduct, which most of our oxygen in the world is produced by phytoplankton. And then also whales, whenever they die, they sink to the bottom, and that's called a whale fall. And they sink to the ocean floor, into the deep waters, and it's pretty bare down in the deep waters. Most of the animals there are scavengers, which means that they will feed on any dead or decaying animal. And there is a whole community. You can see in this picture, this whale is feeding so many different organisms. So the deep ocean relies on whales to sustain it over time. That is all I have for you guys. I hope you enjoyed it. And then are there any questions?
I think we have some questions in the chat. I know I got kicked off at one point, so the earlier questions aren't loading for me.
Okay.
But you should be able to open up the chat and scroll up.
Okay, let's see.
Thank you very much. Yeah, if anyone, if we're going through the questions and you don't hear us hitting yours and you sent one earlier, please send it again. Yeah. I think at one point there were like 4 Taylors on the side. I don't know why I kept picking off that one, but
you were definitely frozen in my screen with your eyes looking up. Yep.
Beautiful. Very cool.
So it looks like Mia is asking they wouldn't try to eat a human, and I'm assuming you're referring to the orcas. So they don't view humans as, or is that Mia? No, okay. They will not view a human as food. They are the apex predators. They will eat sharks. They will eat other marine mammals, but they wouldn't actively attack a human. No. Do you know how much food baleen whales have to eat a day? So they have to eat a lot. They have to consume, I don't know the exact number, so I can't quantify it for you, but they have to eat a lot of phytoplankton a day. Since their prey is really tiny, it is energy rich, but they still have to eat a lot. Yes, Andrea got it, a lot, like gallons, yeah. So whales and manatees, they are both marine mammals. I'm sure in the lineage they are related some way. I don't know how though exactly.
What was the question?
Are whales related to manatees? How so?
I think that we did discuss this earlier when we were going over the presentation, that they branched off somewhere around the same time, like the cetaceans and sirenians branched off separately. And then there were the, what's the one for the otters? Carnivora? Yeah. Because
I know manatees, they're related to elephants.
Yes.
Right? Yeah.
Yeah, they branched off before the cetaceans and the carnivora, because all of the polar bears and sea otters and all of the cetaceans are descendants of like a wolf-like creature.
Sorry, there's a lot of, can you hear me? Let's see. Okay, my question is in 3 parts. My name is Briana Coulter.
I think you could skip that last part.
My quest is to figure out if there are organic contaminants in baleen. That's what I want to know.
Yeah, did you state your actual hypothesis and what you hope your results will be?
Yeah, I think I did in the beginning, but I'll do it again. But yeah, so we are trying to quantify organic contaminants from baleen, and we're hoping that there is, we're hoping to find a temporal, so a time scale, or also a spatial scale. So if there's any variation based off of the time of year or even decades. So our data, we're going to have information from the 60s whenever all of these contaminants were being used, up until the 90s after they were banned, to see if there's any variation in their concentrations. And then also where they're at, if they're by an industrial area, if there's higher concentrations and that kind of thing. Okay, so yes, so the baleen comes from, so this is a good question. So where does the baleen come from? Is it from the dead whales hunted by the Inuit community? Yes, 100% correct. You can't get baleen from a live whale. So the subsistence hunts are very controlled. There's all kind of government agencies there whenever they do land a whale, and all kinds of data is taken, and baleen plates is one of them. So yeah, that was a good question.
I think most of what's left are just people saying thank you. I did have a question myself though. Do you have like an expectation for, based on the specific species and like how they feed and where you're getting the plates from, do you have an expectation for like what organic pollutants are going to be like higher levels?
Yeah, so based off of like the preliminary like literature research that I've done, the like PCBs, so for the more recent PCBs and HCHs, they are most likely going to be the higher concentrations for like the 90s, and DDTs are going to be less. But if we take it back into the 60s, the DDTs are going to be much higher. So yeah, and then there's also been like long-term scale studies based off of the blubber plugs and everything, and they have shown decreases in the concentrations since all of the contaminants were banned, which is reassuring.
Very cool. I'm seeing lots of thank yous. So again, thank you so much, Briana, for doing this. Really appreciate it. It was really interesting. Learned a ton about your project and baleen whales in general. Oh, we have one more question. What harmful effects do these contaminants have on the animals?
So they can disrupt their endocrine system. They can so that's like their nervous system. They can disrupt their reproduction, that kind of thing.
Yeah, we don't want those chemicals in them, and we don't want it in us either. I think the Inuit communities are having issues with higher levels of contaminants because they still eat them.
Yeah.
It's not good. If you do come up with any questions later on, or if we missed any of your questions, feel free to email us at MEEC, MEEC at nova.edu, and we'll be sure to get back to you on it. I will send it right over to Briana if I cannot answer it. Otherwise, thank you so much for coming today, and thank you, Briana, for doing this presentation. I really appreciate it. If you are all free, we are still doing this webinar series every Saturday from here until we open, probably. Every Saturday from 1 to 2 p.m. Next Saturday on the 13th, I believe it is, our very own education coordinator, Carly Todd, is going to be coming in and giving us a awesome presentation on mangrove ecosystems. So that's very important for us down here in South Florida. Our mangroves are a vital part of nursery habitat. They protect our coasts from all of those hurricane storm surges, and they are struggling. So she will give us an overview about all the ecology and all the important bits and pieces and how we can help conserve those mangrove ecosystems. So thank you very much. If you missed the beginning of this, it is recorded. It is also kind of on our Facebook page. I think the reason that I kept freezing and getting kicked off is because it did not want to live stream to our Facebook page. So if you missed any of it, or if you want to share it with anyone else, go to our YouTube page. That'll be MEEC at Carpenter House on YouTube, and you'll be able to find the full series there. Otherwise, thank you guys. Thank you for coming. Thank you for presenting, Briana, and stay safe out there, everyone.
Bye. Thank you.